Hello, today I am going to discuss the female gametophyte. Female gametophyte is nothing but embryo sac. Here, the embryo sac, the formation of embryo sac takes place in the oval in the part of nucellus. The embryo sac, it form in the nucellus. Here, before we are going to study the female gametophyte formation, the one thing we must know that Megasporogenesis. Megasporogenesis. In Megasporogenesis, the microspore mother cell that is diploid in condition, and that diploid microspore mother cell divides meiotically and produce 4 megaspores, and these megaspores are haploid in condition. The megaspore mother cell divides meiotically and produce 4 megaspores, in which the 3 megaspore, it is the microphylar region and this is the chalazal region. The 3 megaspores, they are present at the microphylar region. They get degenerate. And the only one megaspore that is present at the chalazal region, and that become embryo sac. Today we are going to learn how the functional megaspore become embryo sac. Here it is the functional megaspore, and that functional megaspore is having the 1 nucleus, and the 3 nuclei or the 3 megaspores, they are degenerated megaspores. They are present at the microphylar region, and the 1 functional, it is the functional megaspore, and that is present at the chalazal region. Here this functional megaspore divides mitotically. This functional megaspore divides mitotically, in which the mitotic division takes place in a 2 process or 2 stages. These are, the first one is karyokinesis, and the second one is cytokinesis. The karyokinesis means here the only nucleus is divides. After the completion of karyokinesis, the cell wall, cytokinesis takes place, and it means the cytoplasm divides. Here the functional megaspore divides mitotically. Only the nucleus of the functional megaspore divides, and it produces the 2 nuclei. Here the 1 nuclei divides and to produce 2 nuclei. This is the chalazal region and this is the microphylar region. This is the first mitotic division. In first mitotic division, the 1 nucleus divides and produce 2 nuclei, and these 2 nuclei are moved towards to the opposite pole. Which are the opposite poles? The one is chalazal pole and another one is microphylar pole. Here the one cell has the 2 nuclei, that's why it is called 2 nucleate embryo sac. 2 nucleate stage of embryo sac. After that, the again mitotic division takes place. Here the first mitotic division takes place. Again the mitotic division takes place. The second mitotic division takes place. In this also only nucleus is these two nuclei get divides. Here it is the cell. The one nuclei is present at the chalazal region that get divides mitotically and to produce two nuclei. The one nuclei is present at the microfilar region. It divides mitotically and produce two nuclei. This cell has totally 4 nucleus. 1, 2, 3, 4. This is the chalazal region and this is the microfilar region. Now the cell has 4 nucleus. That's why it is called 4 nucleate embryo sac. 4 nucleate embryo sac. Next, after that, again the third mitotic division takes place. Third time the mitotic division takes place. In this also the nucleus will get divides and there is no cytoplasm get divides. Here the nucleus get divides and this is the cell, this is chalazal region and this is the microfilar region. This is the microfilar region and here the two cells they are present at the chalazal region. They get mitotically divides and produce and they become mitotically divides means they become 4 nuclei. And the 2 nuclei present at the microfilar region they get divided mitotically and they become 4 nuclei. Totally the cell has 8 nucleus. Now the stage is called 4 nucleate stage or 4 nucleate embryo sac. 8 nucleate embryo sac stage is 4. Now the mitotic division, the nuclear division will stop here. Totally the 3 mitotic divisions takes place in a nucleus. Here the first mitotic division, the nucleus divides and produce 2 nuclei. Again the second mitotic division takes place. Only in a nucleus it becomes 4 nuclei and the third time the mitotic division takes place in that nucleus and the 4 nuclei get divides into 8 nucleus. And later this stage the cell it is the chalazal region and it is the microfilar region. The 3 cells they are present at the chalazal region and the one cell, here the one cell it move towards at the center of the cell. The one cell comes at it move towards the center of the cell. And here the 4 cells are present at the microfilar region. The 3 cells are present at microfilar region and the one cell from the microfilar region it move towards the center of the cell. Okay, the 3 cells they are present at chalazal region and the 3 cells are present at microfilar region and the 2 cells they are present at the center of the cell. Now it become like this. This is the cell, this is the chalazal region and this is the microfilar region. Here the nucleus get divides mitotically. After that the cytokinesis will be takes place. Cytokinesis means in which the wall formation will be, cell wall formation will be takes place. Here the 3 cells are present, 1, 2 and 3. There are 3 cells are present at the chalazal region and the 3 cells. Here after the cytokinesis takes place means the wall is formed around that nucleus. The wall is formed around that nucleus. Okay. Then Here the three cells are present at the microfilar region. The one nucleus is here. The wall is formed. The one nucleus is here. Also wall form takes place and the one nucleus here, and also the wall is formed. And the two nuclei are again remains in the center of this cell. Now, how many cells are present in a single cell? Here, in chalazal region, there are 3 cells are present. 1, 2, 3. There are 3 cells are present at the chalazal region, and those cells are called antipodal cells. Those cells are called antipodal cells. And next, here the 2 nuclei are present at the center of this cell. The one nucleus is present at the microfilar end or the microfilar region, that is called egg cell. And the 2 nuclei, they are present at the microfilar region. Those are called synergid cells. Synergid cells. Here the one is egg cell and the 2 are synergid cells. These 3 cells are also known as egg apparatus. Egg apparatus. Then the 2 cells or the 2 nuclei are present in this cell. Those nuclei are called polar nuclei. Polar nuclei. Why these nuclei are called polar nuclei? Because of the one nuclei or each nuclei, they are come from the each pole. The one is comes from the chalazal end and the one is comes from the microfilar end. That's why these 2 nuclei are called polar nuclei. And next, here this is the embryo sac. This embryo sac is known as 7 cell 8 nucleate stage. 7 cell 8 nucleate stage of embryo sac. Next, here 7 cells. This entire cell is called central cell. This entire cell is called central cell. Then, how many cells are present in that embryo sac? Antipodal cells. Here, antipodal cells. How many antipodal cells are present in chalazal region? There are 3 antipodal cells. Then, how many egg cells are present in microfilar region? There is only 1 egg cell. One is egg cell, and the two are synergid cells. Synergid cells, synergids. 2 are synergids, and this entire cell is called central cell. That entire cell is called central cell. Central cell, how many central cell is present? Only 1 central cell is present. Totally 3, 4, 5, 6, 7. 7 cells are formed in a embryo sac. That's why it is called 7 cells. How many nucleus are present in that embryo sac? Here the antipodal cells 1, 2, 3. These 3 antipodal cells are having 3 nucleus. 3 nucleus, and the egg cell. The egg cell is having 1 nucleus, and the 2 synergids are having the 2 nucleus, and the central cell, it has 2 polar nuclei. The central cell has 2 polar nuclei. Now, how many nuclei are present in this embryo sac? 3, 4, 5, 6, 7, 8. Totally 8 nuclei are present in embryo sac. That's why this stage is called 7-celled 8-nucleate stage. 7-celled 8-nucleate stage. Then the ploidy level of this embryo sac. The antipodal cells are always haploid in condition. The polar nuclei are always haploid in condition. And next, the egg cell is also haploid in condition, and the synergid cells are also haploid in condition. This is the embryo sac. 7 cells, 8 nucleate stage of embryo sac. Here, one functional megaspore ಇದ್ದಿದ್ದು ಒಂದು ಮೈಟೋಟಿಕ್ ಡಿವಿಷನ್ ಆಗಿ ಇಲ್ಲಿ ನ್ಯೂಕ್ಲಿಯಸ್ ಮಾತ್ರ ಡಿವಿಷನ್ ಆಗ್ತಿತ್ತು. ಈ ನ್ಯೂಕ್ಲಿಯಸ್ ಡಿವಿಷನ್ ಆಗಿ 2 ನ್ಯೂಕ್ಲಿಯಸ್ ಆಗಿ ಫಾರ್ಮೇಷನ್ ಆಗ್ತದೆ. ಒಂದು ನ್ಯೂಕ್ಲಿಯಸ್ ಇದ್ದಿದ್ದು ಚಲಾಸಲ್ ರೀಜನ್ ಸೈಡ್ ಮೂವ್ ಆಗ್ತೈತಿ, ಅಂಡ್ ಅನದರ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಇದ್ದಿದ್ದು ಮೈಕ್ರೋಫೈಲರ್ ರೀಜನ್ ಸೈಡ್ ಮೂವ್ ಆಗ್ತದೆ. ಒಂದು ಸೆಲ್ ಒಳಗಡೆ 2 ನ್ಯೂಕ್ಲಿಯಸ್ ಪ್ರೆಸೆಂಟ್ ಇರೋದ್ರಿಂದ ಈ ಸೆಲ್ ಅನ್ನು ನಾವು 2 nucleate stage or 2 nucleate embryo sac ಅಂತ ಕರೀತೇವೆ. ಆಫ್ಟರ್ ದ್ಯಾಟ್ ಮೈಟೋಟಿಕ್ ಡಿವಿಷನ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಒಳಗಡೆ ಅಗೇನ್ ಮೈಟೋಟಿಕ್ ಡಿವಿಷನ್ ಆಗ್ತದೆ. ಅಂಡ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಇದ್ದಿದ್ದು ಏನಾಗ್ತಾವೆ ಮತ್ತೆ ಡಿವೈಡ್ ಆಗ್ತಾವೆ. 2 ನ್ಯೂಕ್ಲಿಯಸ್ ಇದ್ದಿದ್ದು ಏನಾಯ್ತು 4 ನ್ಯೂಕ್ಲಿಯಸ್ ಆಗಿ ಫಾರ್ಮೇಷನ್ ಆಯಿತು. ಸೊ ಆ ಸ್ಟೇಜ್ ಅನ್ನು ನಾವೇನಂತ ಕರಿತೀವಿ 4 nucleate embryo sac ಅಂತ ಕರಿತೇವೆ. 2 ಚಲಾಸಲ್ ರೀಜನ್ ಸೈಡ್ ಅದಾವು, ಇನ್ನು 2 ಏನದಾವು ಮೈಕ್ರೋಫೈಲರ್ ರೀಜನ್ ಸೈಡ್ ಅದಾವು. ದೆನ್ ಅಗೇನ್ ಈ ನ್ಯೂಕ್ಲಿಯಸ್ ಒಳಗಡೆ ಏನಾಗ್ತೈತೆ ಅಂದ್ರೆ ಮತ್ತೊಂದ್ಸರಿ ಮೈಟೋಟಿಕ್ ಡಿವಿಷನ್ ಆಯ್ತು. ಟೋಟಲಿ 3 times ಮೈಟೋಟಿಕ್ ಡಿವಿಷನ್. ಒಂದು ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್ ಫಾರ್ಮೇಷನ್ ಆಗಬೇಕಾಗಿತ್ತು ಅಂದ್ರೆ 3 ಸರಿ ಏನಾಗ್ತೈತೆ ಅಂದ್ರೆ ಮೈಟೋಟಿಕ್ ಡಿವಿಷನ್ ಆಗ್ತದೆ. ದೆನ್ ಇಲ್ಲಿ ಒಂದ್ಸರಿ ಮೈಟೋಟಿಕ್ ಡಿವಿಷನ್ ಆಗಿ ಈ 2 ನ್ಯೂಕ್ಲಿಯಸ್ ಇದ್ದು ಡಿವೈಡ್ ಆಗಿ 4 ಆಗ್ತಾವು. ಮೈಕ್ರೋಫೈಲರ್ ರೀಜನ್ ಸೈಡ್ ಇರುವಂಥ ನ್ಯೂಕ್ಲಿಯಸ್ ಡಿವೈಡ್ ಆಗಿ 8 ನ್ಯೂಕ್ಲಿಯಸ್ ಫಾರ್ಮೇಷನ್ ಆಗ್ತವೆ. ಆಮೇಲೆ 2 ಸೈಡ್ನಿಂದ ಒಂದೊಂದು ನ್ಯೂಕ್ಲಿಯಸ್ ಏನಾಗ್ತೈತೆ ಅಂದ್ರೆ ಸೆಂಟ್ರ್ ಒಳಗಡೆ ಬರಲಿಕ್ಕೆ ಮೂವ್ ಆಗಲಿಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಆಗ್ತದೆ. ಲೇಟರ್ ಇಲ್ಲಿಗೆ ನ್ಯೂಕ್ಲಿಯಸ್ ಡಿವಿಷನ್ ಸ್ಟಾಪ್, ನ್ಯೂಕ್ಲಿಯಸ್ ಡಿವೈಡ್ ಆಗೋದು ಸ್ಟಾಪ್ ಆಗ್ತದೆ. ನ್ಯೂಕ್ಲಿಯಸ್ ಡಿವಿಷನ್ ಸ್ಟಾಪ್ ಆದಮೇಲೆ ಸೈಟೋಕೈನೆಸಿಸ್ ಡಿವಿಷನ್ ಆಗಲಿಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಆಗ್ತದೆ. ಸೈಟೋಕೈನೆಸಿಸ್ ಒಳಗಡೆ ಏನಾಗಬೇಕು ಸೆಲ್ ವಾಲ್ ಫಾರ್ಮೇಷನ್ ಆಗಬೇಕು. ನೆಕ್ಸ್ಟ್ ಸೆಲ್ ವಾಲ್ ಫಾರ್ಮೇಷನ್ ಆಗುವಾಗ ಚಲಾಸಲ್ ರೀಜನ್ ಸೈಡ್ ಏನದಾವು 3 ನ್ಯೂಕ್ಲಿಯಸ್ ಅದಾವು. ಸೊ ಆ 3 ಸೆಲ್ಸ್ ನ್ಯೂಕ್ಲಿಯಸ್ಗೆ ಏನಾಗ್ತೈತೆ ಅಂದ್ರೆ ಸೆಲ್ ವಾಲ್ ಫಾರ್ಮೇಷನ್ ಆಗ್ತಾವೆ. ಆಮೇಲೆ ಆ 3 ಸೆಲ್ ಇದನ್ನ ನಾವು ಆಂಟಿಪೋಡಲ್ ಸೆಲ್ಸ್ ಅಂತ ಕರಿತೇವೆ. ಇನ್ನು 3 ಸೆಲ್ಸ್ ಏನದಾವು ಮೈಕ್ರೋಫೈಲರ್ ರೀಜನ್ ಸೈಡ್ ಪ್ರೆಸೆಂಟ್ ಅದಾವು. ಅದರೊಳಗಡೆ ಒಂದು ಸೆಲ್ ಅನ್ನ ನಾವು ಎಗ್ ಸೆಲ್ ಅಂತ ಕರಿತೇವೆ, ಇನ್ನು 2 ಸೆಲ್ ಅನ್ನ ಸೈನರ್ಜಿಡ್ ಸೆಲ್ ಅಂತ ಕರಿತೇವೆ. ಈ ಟೋಟಲ್ ಎಗ್ ಸೆಲ್ಲು ಸೈನರ್ಜಿಡ್ ಸೆಲ್ ಎರಡನ್ನೂ ಸೇರಿ ನಾವು ಎಗ್ ಅಪ್ಯಾರಟಸ್ ಅಂತಾನೂ ಕರಿತೇವೆ. ನೆಕ್ಸ್ಟ್ ಇಲ್ಲಿ 2 ಸೆಂಟರ್ ಒಳಗಡೆ ಪ್ರೆಸೆಂಟ್ ಅದಾವಲ್ಲ ನ್ಯೂಕ್ಲಿಯಸ್ಸು, ಆ 2 ನ್ಯೂಕ್ಲಿಯೈನ ನಾವು ಪೋಲಾರ್ ನ್ಯೂಕ್ಲಿಯೈ ಅಂತ ಕರಿತೇವೆ. 2 ಪೋಲ್ದಿಂದ 2 ಎಂಡದಿಂದ ಬಂದಿರುವಂಥ ನ್ಯೂಕ್ಲಿಯೈನ ಪೋಲಾರ್ ನ್ಯೂಕ್ಲಿಯೈ ಅಂತ ಕರಿತೇವೆ. ಈ ಟೋಟಲ್ ಸೆಲ್ ಅನ್ನು ನಾವು ಸೆಂಟ್ರಲ್ ಸೆಲ್ ಅಂತ ಕರಿತೇವೆ. ನೆಕ್ಸ್ಟ್ ಈ ಸ್ಟೇಜ್ ಅನ್ನ ನಾವು 7-celled 8 ನ್ಯೂಕ್ಲಿಯೇಟ್ ಸ್ಟೇಜ್ ಅಂತಾನು ಕರಿತೇವೆ. ಯಾಕೆ 7-celled 8 ನ್ಯೂಕ್ಲಿಯೇಟ್ ಸ್ಟೇಜ್ ಅಂತ ಕರಿತೇವೆ ಅಂದ್ರೆ ಈ ಒಂದು ಸೆಲ್ ಒಳಗಡೆ ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್ ಒಳಗಡೆ 7 cells ಪ್ರೆಸೆಂಟ್ ಅದಾವೆ. 7 cells ಯಾವುವು? 3 ಆಂಟಿಪೋಡಲ್ ಸೆಲ್ಸು, 1 ಎಗ್ ಸೆಲ್ಲು, ಇನ್ನು 2 ಸೈನರ್ಜಿಡ್ ಸೆಲ್ಸು, ದೆನ್ 1 ಸೆಂಟ್ರಲ್ ಸೆಲ್. ಟೋಟಲಿ 7 cells ಆಯಿತು. ಇನ್ನು 8 ನ್ಯೂಕ್ಲಿಯೇಟ್ ಸ್ಟೇಜ್ ಅಂತ ಕರಿತೇವೆ. ಯಾಕೆ 8 ನ್ಯೂಕ್ಲಿಯೇಟ್ ಸ್ಟೇಜ್ ಅಂತ ಕರಿತೇವೆ? ಈ ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್ ಒಳಗಡೆ 8 ನ್ಯೂಕ್ಲಿಯಸ್ ಅದಾವೆ. 1 2 3 ಆಂಟಿಪೋಡಲ್ ಸೆಲ್ಸ್ ಒಳಗಡೆ 3 ಏನಾಯ್ತು ನ್ಯೂಕ್ಲಿಯಸ್ ಆಯಿತು. ದೆನ್ ಎಗ್ ಸೆಲ್ ಒಳಗಡೆ 1 ನ್ಯೂಕ್ಲಿಯಸ್ ಐತಿ. ಸೈನರ್ಜಿಡ್ ಸೆಲ್ಸ್ 2 ಅದಾವು. 2 ಸೆಲ್ಸ್ ಒಳಗಡೆ 1 ನ್ಯೂಕ್ಲಿಯಸ್ ಇರೋದ್ರಿಂದ 2 ನ್ಯೂಕ್ಲಿಯಸ್ ಆಯಿತು. ಸೆಂಟ್ರಲ್ ಸೆಲ್ ಒಳಗಡೆ 2 ನ್ಯೂಕ್ಲಿಯೈ ಇದಾವೆ. ಅವೆರಡು ಯಾವುವು? ಪೋಲಾರ್ ನ್ಯೂಕ್ಲಿಯೈ. ಸೊ ಟೋಟಲ್ 8 ನ್ಯೂಕ್ಲಿಯಸ್ ಆಯಿತು. ಸೊ ಈ ಸ್ಟೇಜ್ ಅನ್ನು ನಾವು 7-celled 8 ನ್ಯೂಕ್ಲಿಯೇಟ್ ಸ್ಟೇಜ್ ಅಂತ ಕರಿತೇವೆ. Here the one functional megaspore that is present at the chalazal region, and that functional megaspore become embryo sac, 7-celled 8 nucleosac, 8 nucleate stage embryo sac is formed, and that process is called female gametogenesis. The process of formation of female gametophyte or embryo sac is called female gametogenesis. In later, I will draw the diagram. I will draw the mature embryo sac. Mature embryo sac. Here it is the embryo sac. The 3 cells, they are present at the chalazal region. This is the chalazal region. Chalazal region or chalazal end region. And next, the 3 cells, they are present at the chalazal region. Those cells are called antipodal cells. Those are called antipodal cells. Next, the one is large cell that is present at the microphylla region. This is the microphylla region. The one is large cell, and that large cell is called egg cell. That large cell is called egg cell or egg. The 2 cells, they are present at the adjacent of that egg cell. Those cells are called, these 2 cells, 1 and this is 2. These 2 cells are called synergid cells. These 2 cells are called synergid cells. These 2 cells are also called the 3 cells, egg cell, and these 2 synergid cells are called egg apparatus. Egg apparatus. And next, here it is the central cell. It is the central cell. Before going to fertilization, these 2 polar nuclei get combined and they become secondary nucleus. Secondary nucleus. In 7-celled 8 nucleate stage, here the 3 are antipodal cells and the central cell has 2 nuclei, and those nuclei are called polar nuclei. Polar nuclei. These are antipodal cells at the chalazal region. It is the microphylla region. The 1 egg cell is present and the 2 are synergid cells are present, and this is the central cell. Before going to fertilization, these 2 polar nuclei get fused and that become secondary nucleus. And next, here the synergid cells, they are having a finger-like projection. A finger-like projection, and that finger-like projection or a finger-like tissue is called filiform apparatus. Filiform tissue or filiform apparatus. A finger-like projection is present in a synergid cell, and that is called filiform apparatus, and this filiform apparatus It helps the pollen grain, pollen tube. It helps the pollen tube. When the fertilization will be takes place, on that time the Filiform apparatus, it helps to pollen tube where the male gametes get produced. Then here it is the central cell. ಮ್ಯಾಚೂರ್ ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್ ಒಳಗಡೆ ಚಲಾಸಲ್ ರೀಜನ್ ಸೈಡ್ 3 ಸೆಲ್ಸ್ ಏನಿರ್ತಾವೆ. ಸೊ ದೋಸ್ 3 ಸೆಲ್ಸ್ ಅನ್ನ ನಾವು ಆಂಟಿಪೋಡಲ್ ಸೆಲ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ದೆನ್ ಮೈಕ್ರೋಫೈಲರ್ ರೀಜನ್ ಸೈಡ್ ಇರುವಂತ ಸೆಲ್ಸ್, ಒಂದು ಲಾರ್ಜ್ ಸೆಲ್ ಅನ್ನ ಎಗ್ ಸೆಲ್ ಅಂತ ಕರಿತೀವಿ. ಇನ್ನು ಎಗ್ ಸೆಲ್ ಬಾಜು ಇರುವಂತ 2 ಸೆಲ್ಸ್ ಅನ್ನ ಸೈನರ್ಜಿಡ್ ಸೆಲ್ಸ್ ಅಂತಾನು ಕರಿತೀವಿ. ನೆಕ್ಸ್ಟ್ ಈ 3 ಸೆಲ್ಸ್ ಅನ್ನ ಎಗ್ ಸೆಲ್, ದೆನ್ 2 ಸೈನರ್ಜಿಡ್ ಸೆಲ್ಸ್ ಅನ್ನ ಸೇರಿ ನಾವು ಎಗ್ ಅಪಾರಟಸ್ ಅಂತಾನು ಕರಿತೇವೆ. ನೆಕ್ಸ್ಟ್ ಇಲ್ಲೇ 8 ನ್ಯೂಕ್ಲಿಯೇಟ್ ಸ್ಟೇಜ್ ಫಾರ್ಮೇಶನ್ ಆದಾಗ ಸೆಂಟ್ರಲ್ ಸೆಲ್ ಒಳಗಡೆ 2 ನ್ಯೂಕ್ಲಿಯಸ್ ಇತ್ತು. ಸೊ ಅವು ನಾವು ಎರಡನ್ನು ಪೋಲಾರ್ ನ್ಯೂಕ್ಲಿಯೈ ಅಂತ ಕರಿತೀವಿ. ಇಲ್ಲೇ ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗೋಕ್ಕಿಂತ ಮುಂಚೆ ಈ 2 ನ್ಯೂಕ್ಲಿಯೈ ಏನಾಗ್ತಾವ ಅಂದ್ರೆ ಕಂಬೈನ್ಡ್ ಆಗ್ತಾವ, ಕೂಡಿ ಬಿಡ್ತಾವ. ಕೂಡಿ ಅದೇ ಸೆಕೆಂಡರಿ ನ್ಯೂಕ್ಲಿಯಸ್ ಆಗಿ ಫಾರ್ಮೇಶನ್ ಆಗ್ತದೆ. ದೆನ್ ಈ ಮೈಕ್ರೋಫೈಲರ್ ರೀಜನ್ ಸೈಡು ಈ ಸೈನರ್ಜಿಡ್ ಸೆಲ್ ಒಳಗಡೆ ಮೈಕ್ರೋಫೈಲರ್ ರೀಜನ್ ಸೈಡ್ ಏನಾಗಿರ್ತೈತಿ ಅಂದ್ರೆ ಫಿಂಗರ್ ಲೈಕ್ ಟಿಶ್ಯೂ ಪ್ರೆಸೆಂಟ್ ಇರ್ತೈತಿ. ಆ ಫಿಂಗರ್ ಲೈಕ್ ಟಿಶ್ಯೂ ಅನ್ನು ನಾವು ಫಿಲಿಫಾರ್ಮ್ ಅಪಾರಟಸ್ ಅಂತ ಕರಿತೇವೆ. ದೆನ್ ವಾಟ್ ಈಸ್ ದ ಫಂಕ್ಷನ್ ಆಫ್ ದಟ್ ಫಿಲಿಫಾರ್ಮ್ ಟಿಶ್ಯೂ ಆರ್ ಫಿಲಿಫಾರ್ಮ್ ಅಪಾರಟಸ್? ಯಾವಾಗ ಪಾಲಿನೇಷನ್ ಆದ್ಮೇಲೆ ಪೋಲನ್ ಗ್ರೇನ್ ಇದ್ದಿದ್ದು ಈ ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್ ಒಳಗಡೆ ಎಂಟ್ರಿ ಆದಾಗ ಈ ಪೋಲನ್ ಗ್ರೇನ್ ಅನ್ನ ಗೈಡ್ ಮಾಡೋದು, ಎಲ್ಲಿ ಮೇಲ್ ಗ್ಯಾಮೆಟ್ಸ್ ಅನ್ನ ರಿಲೀಸ್ ಮಾಡಬೇಕು ಅಂತ ಹೇಳಿ ಗೈಡೆನ್ಸ್ ಮಾಡಲಿಕ್ಕೆ ಈ ಫಿಲಿಫಾರ್ಮ್ ಅಪಾರಟಸ್ ಹೆಲ್ಪ್ ಮಾಡ್ತದೆ. ಅಂಡ್ ದಿಸ್ ಈಸ್ ದ ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್ ಅಂಡ್ ದಿಸ್ ಈಸ್ ದ ಓವರ್ ಆಲ್ ಕಾನ್ಸೆಪ್ಟ್ ಆಫ್ ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್. ಥ್ಯಾಂಕ್ ಯು.